Okay. So hi, my name is Regina Joice Cordy, and I'm a research associate at Emory University. And I'm working with Drs. Mary Galinsky and Alberto Moreno at the Emory Vaccine Center at Yerkes, as well as Dean Jones, Karanapal, and Shuzhao Li here at the Emory Clinical Biomarkers Lab. And so I'll be talking to you today about the work that we've been doing to look at plasma metabolomics in both humans and non-human primates. And this is work that's being done as part of the Malaria Host Pathogen Interaction Center, which Shuzhao introduced earlier. And so this is a consortium that's based at Emory that is using non-human primate models and multi-omic data profiling in order to look at host and parasite changes during the course of malaria disease. And so in addition to the primate work, we're actually also looking at several different field sites and looking at plasma metabolomics in humans infected with malaria. And so, so far, we've looked at and we've collected samples from Colombia, Brazil, and Thailand, and we've already done mass spec on those samples, and we're now in the process of analyzing those and trying to understand what the plasma metabolome looks like during malaria infection. And the reason that we're looking at different areas in the malaria endemic regions is that we're also looking at comparing the different Plasmodium species, including Plasmodium falciparum, Plasmodium vivax, and Plasmodium knowlesi. And so overall, the approach that we're using is trying to understand host factors that may be involved in malaria disease progression. And so one thing that we're actually really interested in is trying to use different, whether it be humans or non-human primate experiments, looking for different metabolites that are altered specifically in malaria infection versus those that are altered in other infectious diseases or other febrile illnesses, so that we can separate out which of these factors are related to the host immune response or overall physiology, as well as those that are specific to malaria. And then within malaria, we're interested in comparing between, as I mentioned, the different Plasmodium species, but we're also interested in comparing the host metabolome as parasites have different ratios of asexual and sexual stages, and also during mild versus severe disease. And overall, one goal would be to identify candidate metabolites that could actually be used as some sort of a prognostic biomarker and that could perhaps identify a certain condition of malaria or help to predict whether an individual is more likely to progress toward mild or severe malaria. And so I'm going to start by talking about the work that we're doing in collaboration with Jetsuman and Rapat at the Mahidol Vivax Research Unit. And so this is work that we're doing looking at Plasmodium falciparum and Plasmodium vivax in individuals in Thailand. And so this is a cross-sectional study that we looked at, and we looked at a number of individuals with Plasmodium falciparum, Plasmodium vivax, actually co-infection of both Plasmodium falciparum and vivax. And then we looked at two, what we're calling control populations. So from the same country, we looked at the plasma metabolome of individuals that were healthy, as well as individuals that had a non-malarial febrile illness. And this category was actually made up of diverse causes, and it included dengue fever, influenza, undifferentiated fever, and autoimmune disease. And so this is a way that was designed so that we could look at both factors that are specific to malaria, but also look at factors that may be more general to different disease conditions. And so we're using the workflow that's very similar to what's been talked about in the previous talks, and Shuzhao also mentioned it at the beginning. So we're using high-resolution metabolomics, and we're using this to measure a large number of metabolites all at the same time. We're using liquid chromatography in an ultra-high-resolution mass spec. And then we're using a number of different bioinformatics methods to extract out the M to Z or mass to charge ratios, which give you the individual profile of specific metabolites. Using these bioinformatics approaches, we're also coming up with putative annotations for these metabolites. And then we're using biostatistical analyses as well as pathway analyses to come up with metabolites and metabolic profiles that appear to be different in the different categories that we're comparing. Finally, as we go into the last stage here, as we start to identify metabolites that appear to be interesting, we'll be using targeted mass spec to confirm the identities of these metabolites. And so this is what we see when we look at a global view of that study that I just described, the cross-sectional study in Thailand. And so what you can see is that we have, let's see, where? Sorry. Okay. So each of these, this principal components analysis, each of these symbols represent one of the different categories that I explained. And so we have Plasmodium falciparum, vivax, and co-infection, as well as the 2 control cohorts. And what you can see is that there's actually a clustering of healthy individuals that are separating out from individuals with all of the other disease conditions. You can also see that the non-malarial febrile illness category is also separating from healthy, which is interesting because these are a variety of different conditions, but they're all separating out from healthy. So we see that there's definitely some general profiles that are changed in those cases. And then as you can see, the malaria cases are even further clustered from those non-malarial febrile illness cases, which is interesting because it looks like there are specific changes in malaria that are separate from the non-malarial febrile illness. And then also we see that the malaria different species are actually overlapping in this analysis. And so we know that there must be a number of different pathways that are in common across the malaria species, as these are found to be overlapping. And so looking at a 2-way hierarchical clustering analysis, we're digging in a little deeper to find out what sort of metabolites appear to be changed in these different groups. And so what you can see here is the samples that I've just described are all along the x-axis here. And this darker green is the healthy individuals, and the red is the non-malarial febrile. And then these are the malaria categories. And you can see that there are 2 major clusters, one with the non-malarial cases and one with the malarial cases. And as we look through this heat map, we can see a number of different profiles that show possibly metabolites that are happening, that are have different patterns to them. And so, for instance, here you can see that there are some metabolites that are enriched in both malaria and non-malarial febrile illness, but that are down in healthy. You can also see that there's a large number of metabolites that are up during malaria and down during the non-malarial cases. And then we also see that in our non-malarial febrile illness category, in a subset of the individuals, there are certain metabolites that are at increased or decreased concentrations. And so we think that these may have to do with some of those specific viral illnesses perhaps that are in that category. And we also noted that there is a specific set of metabolites within the malaria cases that seem to be enriched primarily in Plasmodium vivax. And so this is something that we're actually really interested in focusing on and looking at annotations for so that we can understand what those metabolites might be. But what we've focused on for right now is really just trying to look at the differences between the overall malaria versus the non-malaria. And so what you can see here is that this is an example of one of the metabolites that's enriched in all the illness categories, malaria and the non-malarial febrile illness. And this is, the metabolite is called kynurenine, and this is related to immune signaling. And so this just gives you an idea of the kind of things that you find when we're looking at the global host metabolome, is we see a variety of different factors, especially those involved in the immune response, that are enriched during malaria, but also in a number of other disease conditions. It's also been reported in a wide variety of studies and different analyses that there is a widespread perturbation of lipids during malaria infection. And so this is just demonstrating that we also observe perturbations, and giving you just 2 examples. One is of a lyso-PC, where you can see that in healthy individuals, you have a higher level than in all the other disease conditions. So this is again showing a situation in which there's a specific lipid that seems to be decreased levels during both the malaria and the non-malaria. And this is particularly interesting because we know that there's both reasons for the sake of the parasite using lipids to create membranes, that the parasite might be depleting this lipid, but there's also an immune signaling and immune related component to that as well. And so there could be host and parasite reasons for why there are changes in those levels. We also see that C18 sphinganine is highly enriched in the Plasmodium cases in all of the different categories. And so this is also showing us a different situation in which we see a lipid that's increased specifically more in the malaria cases than in not. And so we can see from looking at analysis like this is a very small cohort, but it's starting to give us a view of the type of things that we can do with plasma metabolomics in patient samples. We can see that malaria and febrile illnesses do have a subset of metabolites that are in common. that seem to be changing during illness states. We also see that, as others have discussed, that there's a perturbation of lipids during malaria. And from these cross-sectional studies, it's not very clear how these relate to clinical disease progression. And as I mentioned, we're really interested in how these metabolites may be used to look at how the progress of malaria infection proceeds. And so, as I mentioned, I'm working with the Malaria Host Pathogen Interaction Center, and we're focused on non-human primate models. And so we then turn to our non-human primate model to look at the dynamic of these metabolites and to try to better understand what clinical changes might be happening alongside these shifts in the metabolome. And so I'll just show some data from our infection model of Plasmodium cotinii and rhesus macaques. And so Plasmodium cotinii in rhesus macaques is a model for Plasmodium falciparum. And so it displays some of the characteristics of falciparum that make it a good model system. These include features such as the vascular sequestration, the antigenic variation, and the severe pathology that goes along with that sequestration, as well as anemia in the red blood cells. And further, using longitudinal follow-up, we can also study not just acute infections, but we can also study the recrudescent and chronic phases of the malaria infection. And the way that we do that is sporozoites are inoculated into these animals, and then as parasitemia rises, parasitemia is the black dots, we get to a point where we treat the animals with a subcurative dose of artemether. At this point, the parasitemia declines, but it is not eliminated. And so that gives us this phase, which we're calling the recrudescent phase, where the animal is working to control parasitemia. And you get rises and falls in parasitemia during that time. Then we drew a line at the point where parasitemia is kept to under 1,000 parasites per microliter or less. And we also note that during that time, we also see hemoglobin start to increase, though it doesn't come back up to baseline levels. So we call that the chronic phase. And so we're interested in looking at these different phases to look at the dynamics of the metabolites across the course of infection. And another thing that we're very interested in is also host variability. And so this is showing the cohort of animals that we studied during that experiment. There were 4 individuals, and you can see that in each of the 4 individuals, we have a different path towards disease progression. So starting at the bottom here, we have an animal that quickly self-controlled parasitemia following 1 subcurative dose. Then we also have 2 animals that were sort of intermediate in terms of taking a little bit longer before they were able to control down to that low parasitemia. And then here at the top, we have an animal that required 4 additional subcurative doses as it was never able to self-control parasitemia during that phase. And so this gives us both a way to look at the longitudinal dynamic, but also to look at what are the host differences that we see during the course of infection. So, of course, we're interested in the immune differences, but also in overall host physiology, and using metabolomics as a way to just start off looking at the differences that we see animal by animal. And so, again, we used the metabolomics methods that I described in the beginning. And what we did was we did a analysis comparing those categories that I explained, the baseline, the acute phase, recrudescent, and chronic. And so what you can see in this heat map, this is showing the statistically significant features that were different between those categories. There were 3 primary clusters. The baseline formed a cluster in which you can see certain metabolites are lower and then increase during malaria, and then we sort of have a vice versa of these lower metabolites. Then we also saw that the acute and recrudescent phase actually cluster together. And that was fairly interesting because we didn't a priori know whether the recrudescent phase would look more like chronic or more like acute. And here we actually see that from a metabolomics view, it looks like that whole section has a very similar dynamic. And so this could have to do with things like the parasite being at higher levels and related to parasite nutritional requirements and or certain things that the immune system is doing at that time. And then finally, the third cluster is the chronic phase, where we do see another shift in the metabolome. And so this is using a pathway analysis software called MummyChug, which was actually designed by Xu Zhao. And what you can see here is this is a way that we compare, using that data, look for globally what are the metabolic pathways that are the most changed across those categories of the infection. And so what you can see, I'll just point out a couple of things, that there are a number of amino acid metabolism pathways here. And of course, as Manuel talked about, and we know that parasites are going to cause dramatic changes in the amino acids due to their need to take up amino acids either by degrading hemoglobin or taking them up extracellularly. And then we also see tryptophan metabolism, which is related to the kynurenine metabolite that I talked about at the beginning. And so we also know that we're identifying pathways that may be related to the immune response. And so this is just to show you the kind of methods that we can use to try to get a global picture of what's happening across the disease conditions. And then finally, what we did is, and we're in the process of really digging into this data and trying to do this for a large number of metabolites, is we're looking at some of the metabolites that appear to be interesting in the human samples. So I talked about the Thai example where, for instance, we were looking at lysophosphatidylcholines, and here is just showing one of these metabolites over the course of infection. And so what's interesting about this is, as we see that this metabolite was at lower levels during the disease conditions in the Thai data, we also see that it falls from its baseline levels in all the animals. But then what's interesting is that the animals differ in their dynamic at which they actually recover. So you can see that these two animals at the bottom have a kind of faster recovery than the ones do at the top. And so we're really interested in trying to associate that with all the cellular data that we're generating on this experiment and really understanding those patterns and how they relate. Just another example here was that C18 sphinganine, and you can see that in all animals at the acute phase, which is the time point 2, you can see that that metabolite is at a detectable level. It was undetectable at baseline in 3 out of the 4 animals and undetectable by the end of the infection for all of them. And you can see that in these bottom 2 animals, it goes up and comes right back down, whereas it stays higher up in the top 2. And so we think this is also something that when we start to compare with the cellular data, we can also look at how this relates to the parasite dynamic as well. And so just to summarize, I showed you some data from a cross-sectional study of humans infected with Plasmodium species, and we could see that there were similarities that we observed between the species. We also noted that there is some distinct plasmolipids associated with malaria, as has been shown in other studies. And then now we're working to really expand this to those other cohort studies that I mentioned at the beginning and try to understand whether we see these patterns in different cohorts. And then also, moving into the experimental work, we're really looking to use the animal models to understand the dynamic of these metabolites, to see how they differ in different phases of infection, and also how they differ across animals to get a better idea of how host variability plays into this whole picture. And so our next steps here are really to analyze this jointly with the clinical and cellular metadata that we have collected on these animals. And this is part of, as I mentioned, a multi-omic consortium. And so from every experiment, we actually have data on different platforms, such as blood transcriptomics and lipidomics. And so we can really go in and work to validate these findings on other platforms. And so we'll be working on that as well. And so I just want to stop here and just acknowledge everyone at the MAPIC Consortium, as well as the Yerkes Primate Research Center, the Emory Clinical Biomarkers Lab, and the Mahidol Vivax Research Unit, as well as the hospital there at the Mahidol University and our funding sources. And I'm happy to take questions.